First, we're going to start off with Reynilda Montez, Dr. Montez, as we call her now, that she's graduated. But she's been working with me for the last couple years on a variety of projects. And today, she's going to really share some work that she's been doing, looking at soil profile characteristics and how they influence yield in a couple fields in Grant County, and then how those also interrelate to soil health parameters and nutrient management. I don't know how much you're going to get into. She has a mountain of data on this project, and she's going to share a little bit with you, but I'll let you take it.
Yeah. Hello. Hello. Can everybody hear me? Is it okay? No? That's okay. Yeah, I have a loud voice, so. Yes, good afternoon. So as Dr. Warren introduced me, I'm Reynilda Monteza. So I am requested to present some of my research for my doctoral study. And I will be talking about how inherent soil properties influences some of the soil health indicators and also crop yield at the field scale. Okay. So let's start. So just to give you an overview. Okay, yeah. So we all know that soil varies from one space and from time to time. And okay, yeah, I have to be very particular with this one. So we know that different soils have different intrinsic limitation because they have different unique inherent properties. So when we say inherent properties, these are properties that are a product of soil forming factors. They are formed for a longer time, so they do not change so easily with management or other natural stressors or disturbances. So let's take, for example, this study here from published in an article in Australian Journal of Soil Research. So they studied how one of the soil properties, which is soil electroconductivity, affect wheat grain yield. And in this study, they used remote sensing, and they saw a relationship that when you have higher electrical conductivity, you actually have lower grain yield. But then the question here is, this electrical conductivity is a property that is a product of both static. Oh, sorry for that. Oh, yeah, sorry. It's my nervousness. I didn't turn it on. So it's a product of both static and dynamic properties, meaning it can be affected by other properties of soil. It could be affected by soil, by salinity, by clay type, by water content, and any other type of properties. But most importantly, is affected by inherent properties. And most of the time, these inherent properties are rarely studied, or there is little accommodation in terms of focusing on research about these inherent properties. And that is because these inherent properties are mostly have an unmanageable constraints in a very limited time. So it takes time for it to be managed. So for agricultural system, which we're in, we wanted to have a cropping every season, twice a year maybe, or once a year. We wanted to manage the soil as much as possible as we can. And that is the reason why most of the study are not really into this type of properties of the soil. But in our research, we wanted to see that we wanted to show that these inherent properties actually affect how soil function naturally. So with that differences in intrinsic limitations, there is an increasing need to identify different management zones within a field. Because of these differences in how soil function or how soil is capable of producing crops, there are different variability in the yield within a field. And so there are a lot of research that has been conducted to identify what are causing this variability in crop yield. And most of the studies that has been conducted are mostly focusing on dynamic properties. So when we say dynamic properties, these are properties that are easily changed with management. In comparison with inherent soil properties, these are properties of soil that does not change easily with management. And that is because, as I've said, they are a product of soil-forming factors. So when we say soil-forming factors, these are like climate, parent material, vegetation, how the land is managed, topography, and time. So they take a long time. But for dynamic soil properties, these are easily changed in management. It could be a natural or anthropogenic disturbances or stressors. And some examples of inherent properties include soil color, texture, clay mineralogy, depth, just among the others. For dynamic, we have bulk density, some fertility parameters like the nutrients and soil organic carbon. And most of these dynamic soil properties are influenced by inherent properties of the soil. So inherent soil property, as I've said, are a result of soil forming factors. And then for dynamic, it's mostly, most of the soil health indicators fall under this category. So it means that most soil health indicators actually is very much affected with any management, just a little or much management in the field. And then these kind of properties are commonly measured mostly on the topsoil. There are some studies that they measured in the topsoil, but mostly they concentrate on the topsoil that are being tilled in agricultural systems. So the top 30 centimeter perhaps, or the top 6 inches. For inherent soil properties, it extends throughout the soil profile. So when you wanted to study the soil, you have to evaluate the morphological characteristic of the whole profile, and that wouldn't change for a long time, because whatever affects that has happened like long before. So it would change, but it will take millennia or thousands of years. And so that's one of the reasons why it's so hard to manage it, because of that, but also it's important to acknowledge its importance in affecting other properties, specifically dynamic soil properties or soil health indicators. So in our study, in my study, we have two objectives. So we wanted to evaluate the relationship between these inherent properties, which is we interpreted as depth to limiting layer, with the spatial variation of crop yield in the field. And then the other one is we wanted to evaluate the relationship of this depth to limiting layer with the surface, with some of the surface soil health indicators. So if some of you are not familiar with soil health indicators, so these are just soil properties that are being measured to evaluate the soil health status. So it could be soil organic carbon, other nutrient, and depending on the concern of the researcher. So in our study, what we did is we collected soil core profile across sites. So we have two sites in Grand County, the Yonker and the Phi site. So we collected different soil profile core samples, and then we determined the morphological characteristic of each profile, wherein we identify the soil color, the soil texture, structure, all other special features that you can see in a profile, the pores, the root growth, redox features, and all of these inherent soil properties are used in order to determine the depth to limiting layer for each soil core profile. So, for example, the clay content, a clay content of more than 35% is considered to be a restriction for some of the soil function or even root growth. And then soil structure, like for example, a massive structure or a prismatic structure or a type of structure that could actually impede how soil function. For example, how water flow, how nutrients is restricted, and all others. And then soil depth, it could be natural stressor or anthropogenic stressor. For example, for natural, a shallow versus a deep soil formation. So you have a very deep soil that even though 2 meters away from the surface, you still have soil and you can't see the bedrock or any rocks at all. When we say deep, when we say shallow, at a very shallow level from the surface, you can already see the presence of bedrock. So those are actually considered to be a limiting layer because plants' root can actually penetrate into that, and that actually limits the nutrient availability in that soil. And also compaction, so it could be natural or anthropogenic compaction. So because of tillage activities, it can cause compaction, especially if you have a very high clay content in the subsoil, and then you always till the soil every season, so that could cause a compaction. And there's also that so-called natural compaction, and that is entirely because of the properties of that soil, mostly because of the clay content or the water content, how water flows in the soil, and all others, or presence of other factors. And then we have dense steel, and then presence of sand and gravel. And then for other features, like for example, redox features, like presence of mottles, glaying. So if you've ever seen a soil that has a very poor drainage, like when you dig into the soil and you see this grayish color, and then you observe that some time of the year there's like water impeded along that profile, so that is actually an indication that there is a limiting layer in that profile, and that could actually affect how your crop functions or how you grow crops in that soil. So all of these inherent soil properties are used in order to determine the depth to limiting layer, which should be at least 10 inches or more in thickness. So just to give you an idea of how it looks, so this is an example of a soil profile where you can see here, this is a presence of sand and gravel. That could be an anthropogenic activity wherein they transfer materials from one place into their farm, and over time it become a part of the soil profile. And then we have here the presence of a very high clay content, as indicated by a very compact soil structure. So if we have a bigger sample of soil, it could actually be a massive structure, but in here, it looks like a prismatic or a very big chunk of a blocky structure. So the structure actually also indicates how the soil can inhibit or impede the root growth or movement in the soil. So those are some of the consideration that we take in determining the depth to limiting layer. So for the result of our study, so in talking about relationship between depth to limiting layer and crop yield, so first let's talk about the Yonker here. So here you can see that the depth to limiting layer is actually influencing the variability of crop yield in this site here. Like for example, in soybean and wheat, so here we have a soybean here, you can see here that when we have a shallow depth to limiting layer, this actually means that at 10 to 30 centimeter, there are some profile that has a limiting layer in that layer of the soil, and that actually means that your crop roots can actually have some hard time penetrating or taking up some nutrients or other constraint in terms of growing. So that limiting layer is limiting how that crop grows and produce. So here we can see that as the shallower the depth to limiting layer is, the lower is the yield for soybean, and as we go deeper for depth to limiting layer, the higher the yield is. The same trend that we see in wheat here, so we have here the same trend wherein the shallower the depth to limiting layer, the lower the yield, and then the deeper the depth to limiting layer, the higher the yield. So this graph here only shows that the variability of this crop yield, the wheat and the soybean yield for this specific site here can actually be explained partly by the depth to limiting layer. As much as 80% of these differences in yield across that field can be explained because of that depth to limiting layer. The other site that we have is the Feist here. So they have soybean, wheat, and corn. So the same trend, we observe the same trend for this site. So the shallower the depth to limiting layer here, the lower the yield, and the same, the deeper the depth to limiting layer, the higher the yield. And that is true for all other crops that has been observed in this site here. Though compared with Yonker, we have a lesser relationship between the depth to limiting layer and the crop yield. So these inherent properties can only explain as much as 60% of the variation in yield. But regardless of that, it's very important to take note that this graph is showing us that the depth to limiting layer, or the combination of all the inherent properties in that soil, is actually affecting how the soil functions and how it actually supports plant growth. So for the relationship between depth to limiting layer and some soil health indicators, so we have here data from FICE. So we only have observed significant relationship with the depth to limiting layer and soil health indicators for one site. For the Yonker site, for some reason, the depth to limiting layer is actually affecting some of the health indicators, but it's not much of a significance, so we're not reporting it here. So I'm only presenting one site here. So here we can see, first let's look at the MWD here, that is the aggregate stability. So it actually tells you how stable your soil is. So when we say aggregate stability, so how your soil particles is forming together, and that actually is affecting how it support root growth or how it support, mechanical in terms of growing crops. So in here, we can see that there is a positive relationship between depth to limiting layer and aggregate stability. What does it mean? It means that the shallower your depth to limiting layer is, the lower your aggregate stability. And the deeper your depth to limiting layer, the higher your aggregate stability. So this one is a tricky one because our initial hypothesis is that when we have deeper depth to limiting layer, we will have lower aggregate stability, because we were thinking then the layer above that depth to limiting layer will have lesser clay, so they will become more coarser in texture, so they will have more sand and silt in it. And knowing sand and silt, it has less capability of binding those particles together, as clay is more capable of doing that. But then in here, we have an opposite result. And so what we're thinking here, the reason for this is that when we have a depth to limiting layer at a very shallow level here, it means that the higher clay, the layer that has a very high clay content, which is in most cases that we have here in our study is more than 50%, 60% clay. So that is a very, very high clay content for any type of crops. And that could actually cause a significant compaction in a way, especially if the soil is tilled for like twice or thrice a year. So with that, what happened is if you have that very high clay content in that layer and you tend to till that soil, so you tend to break up those particles together, and when it broke up, so what happened, because they have these finer particles, there are a lot of clay in it, so they somehow rearrange themselves compared to soil that have coarser particles. So we know that clay has a fine size, right? So when you break them apart, they rearrange themselves in a way that is not naturally, in a natural manner, or in a manner like before with so much pores, for example. So when it rearranges itself, it becomes more compact, it loses its pores, or the presence of pores. So somehow it inhibit the stability of those aggregates. So that's one of the things that we're thinking why we have this kind of trend in this site here. For the other parameters, we have an opposite trend wherein we have a negative relationship between the depth to limiting layer and the parameters like total carbon, nitrogen, phosphorus, and potassium. So what does it mean? It means that when we have a deeper depth to limiting layer, for example, 90 to more than 100 centimeter in your profile, it actually means that you have lesser amount of these parameters. And when you have shallower depth to limiting layer, meaning your limiting layer is almost near the surface of your soil, so it means there are more of this, more amount of this total carbon, nitrogen, phosphorus, and potassium. One explanation that we're thinking of this is because you have the clay content very high, and we all know that clay can actually hold better nutrients and water compared to coarser textured soil. For example, sandy soil or silty soil. So when you have more clay, it actually holds more of the nutrients and more of the water, which is actually causing to have this higher concentration of these nutrients that we have here, like nitrogen, phosphorus and potassium, as well as those total carbon. So those carbon are being protected by those clay much effectively than with sand. So since we have a clay layer, very high clay content layer at the upper layer, this layer are capable of holding this organic matter, transforming it into carbon, and then holding this carbon and reserving it, and that is what must be the reason why we have recorded here a higher concentration when we have a limiting layer at a shallow depth. So that is for the 0 to 5 centimeter soil depth. For the 5 to 15 centimeter, we observe the same trend here for aggregate stability, total carbon, and nitrogen, but we didn't observe the same for potassium and phosphorus. But we have an additional health indicators that has a relationship with depth to limiting layer. So we have here the electrical conductivity and the sodium adsorption ratio, or the SAR, which is an indication of salinity in the soil. So here, the same explanation goes with the aggregate stability, total carbon, and nitrogen. For the electrical conductivity and the SAR here, so we also observe a negative relationship between limiting layer and these parameters. So it means that at deeper limiting layer, you have a lower content or lower concentration, lower value of these parameters. So the takeaway from this study here is that the variability of crop in the field can actually be partly, so just partly, because in reality, there are a lot of factors that can actually affect how plants grow in your soil. And your soil is also affected in itself by different factors, internally or externally. But it's also important to take note that according to the result of our study, as much as 20 to 90% of this variability can actually be explained by the depth to limiting layer, which is a combination of the inherent properties of the soil that we evaluated. And here, just to give importance, among the crop that we observe, the soybean is highly influenced by the depth to limiting layer compared to wheat and corn or sorghum. We didn't present the sorghum here, but that was the case. So one reason for that is that wheat, for some reason, climate is also affecting it heavily, because wheat is harvested during the time where we observe that there is a high precipitation during some years where we harvested the wheat, and that for some reason affected the amount of yield harvested during that time. But it's not necessarily mean that it affected the growth just during the harvest. So we did not present that data because it is very much away from the effect of limiting layer compared to the effect of climate with this yield. And also lastly, we observe more negative relationship between depth to limiting layer and some soil health indicators. But this relationship is not consistent with depth and also with site. So what we're thinking is, if we have more sites which are more diverse in terms of inherent properties, maybe we will have more much better data that we can show that the depth to limiting layer is really affecting how your soil function and how it support plant growth. That's it. Thank you. Any questions?
Thanks, Reynilda. Does anybody have any questions for Reynilda? Anybody? Okay. Well, one thing I'll point out, she's been working on this for the last 3 years, and one of the reasons we did this is because there's a lot of discussion about soil health and carbon content of soils and how those things drive yield. And what this data shows is that the inherent soil properties that you have that you cannot change dictate a lot of what your yield potential is. And I get a lot of questions about how we can change that. Well, we can't change how much clay you have in the subsoil, how deep it is, unless you want to get a lot of money involved in it. And it's interesting too, is as we look at trying to monitor these near surface characteristics, there's a lot of stuff to unravel in that. But you know, with the nutrients, if you have low yielding areas, because of the inherent properties, you're going to accumulate nutrients in those, like Reynilda found, and then you may or may not see accumulations in organic matter, because if you've got less clay in those surfaces, they're not going to hold that organic matter. And we can get a real deep dive into what Reynilda is working on, but I don't want to bore you with too much soil stuff. But thanks, Reynilda. Next we have Mary Foltz. I was really lucky to go to a seminar that Mary presented a couple years ago when she first started at faculty in the Civil Engineering and Environmental Engineering department, because we don't get to hang out with those folks very much in Ag Hall, but she came over to Ag Hall and was talking about her research interests related to greenhouse gas emissions, and all things related, and so I've been able to collaborate with her on a variety of things because she has skills that I do not have. I just measure stuff for a really long time and generate a lot of numbers, but Mary can make a lot of sense out of that. And so she's going to talk about life cycle assessments in agriculture, and as we move into this, you know, future where we're going to see a lot of money poured into trying to reduce our carbon footprint and mitigate climate change in agriculture, this kind of discussion is going to be very important as we try to navigate the future as I see it. So I'll give it to Mary.
Can you hear me all right? Okay. I think I have a real pocket. It's the downside to women's clothing. All right, so as Jason said, I'm Mary Foltz. I'm in environmental engineering, and I'm hoping that I can take the crop school to heart and really teach you guys something today, and that is going to be life cycle assessment. So has anyone heard of life cycle assessment or sustainability or anything like that? It's kind of like the buzzword or buzzwords right now. And so I'm hoping to put that into context of agricultural systems and how that can be applied and how it likely will be applied in the future. Just a moment. All right, there we go. So life cycle assessment is really a tool or a framework that's going to be applied to any system or product, and you have to consider the entire life cycle as inherent in its name. And so you want to look at from the creation of it or the birth, the cradle, all the way to the grave and whenever the product or system is disposed of, if it's ever disposed of. And so this is a slide that I stole from one of my good friends at the University of Kentucky because she's really an expert in this quantitative sustainable design and looking at life cycle assessment. And she's predominantly worked in the fields of environmental engineering and water resources, sanitation engineering, that type of thing. But I was like, you know, with all of this excitement over sustainability in ag, I think it could be really relevant in these systems as well. And so the idea behind an LCA is that you start with your goal and scope, where you set the framework, you say what system you're looking at, what are the bounds, what are your assumptions and limitations. So that's the first step. And then the next step is inventory analysis. So that's where you take and make a list of all of the inputs going into your system or your product, and then turn those into emissions to the environment. So for every single piece of fertilizer that goes in, you have some kind of emissions coming out or some sort of output. And then impact assessment takes those outputs, those emissions, and translates those into environmental impacts. Those environmental impacts could be greenhouse gas emissions, climate change, that's the big ones that folks are talking about, also eutrophication and nutrient leaching, fossil fuel depletion, that's a big one. So there's a lot of different categories that we can look at. And then the final step is interpretation, where you take all of these pieces together and try to come up with some sort of recommendation. we like to show arrows between all of these processes and golden scopes sort of surrounding it because they're all interconnected, and it's not always a stepwise process. Sometimes you have to go back and revisit previous steps. So that's the overarching framework for an LCA. Now I'm going to go through kind of step by step and talk about how this can be applied in agriculture, and then give you an example from some work that Jason and I did recently. So the first step, goal and scope. These are the list of things from ISO standards, and essentially it's defining the who, what, when, where, why. That's what I tell my students. What are the systems that you're considering? What is the functional unit? And that's a terminology that's probably only used in LCA, but I'll talk about it a little bit today. Setting a system boundary and so on. And so, for example, in agriculture, you might say that your system considered is your agricultural field. And you might consider that in comparison to your neighbor's agricultural field or something like that. The functional unit is basically how we're going to normalize this information. And so in agriculture, that's often either done on an area basis, or in our opinion, we think it should be per unit crop produced, so that you are incentivizing those who have the managements that are producing the best yields. And so they're going to essentially decrease their total environmental impacts based on the crop that they are yielding. And then for system boundary, that's where it gets complicated, and I'm going to hopefully go into that a little bit with you here today to show you just how complicated it gets and why we have to make so many assumptions. Oh, there we go. Okay, so as you are familiar, agricultural systems are managed in so many different ways, and these are just a few. And I'm going to only present to you just a snapshot of what these look like as you add them in. So let's start with fertilizer or nutrient amendments. So in my work, I do a lot of the denitrification process and nitrification process and looking how nitrogen is cycling in these soils. So fertilizer is applied in some of these different forms. We have nitrate, ammonium, and once it enters the soil in that reactive form, it can be easily transformed or lost from the system. And actually, 70% of the nitrogen that enters the agricultural system is lost before it even reaches that crop. And so that's a problem. And we've addressed that by coming up with more efficient fertilizers or talking about the right time and the right place and things like that. But if it's not perfectly aligned, it could end up going through denitrification process. So that's where nitrate is reduced through all of these other compounds here. And the big one that I talk about a lot in my work is nitrous oxide. And the reason that we care about nitrous oxide is because that is a potent greenhouse gas. So it's even worse than carbon dioxide. Just it's more powerful, essentially. And all of these ones here on the right, those are all gases. And so before they can be fully reduced all the way to dinitrogen, they might be released as nitric oxide or nitrous oxide, or just released as dinitrogen. And we don't want that. You know, you want all of that money you spent on the fertilizer to go into your crops and actually give you good yields. We don't want it denitrifying and being released back into the environment. That's not the goal. And then there are other processes, like nitrification is the reverse process happening here, where we could go back to nitrate. Maybe then it would enter up into the crops, or maybe it would denitrify. And so this is a very complicated system here. And all of this is just talking about the direct soil emissions. So what is coming from the soil directly. And also just talking about nitrous oxide and nitrogen emissions. So we have these direct soil emissions from fertilizer. That's all I'm presenting here. But that's not the only greenhouse gas emissions associated with fertilizer use. There's also pre-field emissions, which is the production of the fertilizer. We have the Haber-Bosch process, which uses steam methane reforming to get our hydrogen that we can then combine with the dinitrogen in the atmosphere, break those bonds with a lot of high energy, high pressure processes. These emit a lot of carbon dioxide. And so this intensive process here is going to also produce these greenhouse gas emissions, or GHG. And And then we have things post-field. So not what happened before it got to your field, not what happens in your field, but let's say those nutrients, instead of being emitted to the atmosphere, they're leached out or they run off. They can go through these soil processes downstream and also emit that nitrous oxide. And so this would be post-field. And so are you starting to see how this is getting complicated? And we wonder where to set our system boundary. And that was only talking about fertilizer. Let's introduce irrigation. So irrigation is going to modify these soil processes. Namely, we say that it's going to increase the denitrification because more water is going to make an anaerobic system. And anaerobic systems like to denitrify and make that nitrate disappear. And so that's possibly happening. And so if we add in irrigation to this system, we have production of energy for irrigation, like for pumping your irrigation water. We also have the introduction of this water and how that's going to influence your nitrous oxide emissions. And things are getting messy, and we only have 2 managements here and only have greenhouse gases considered. And so in life cycle assessment, we always have to set our system boundary and say, this is the only area we're looking. Everything else we're going to ignore because we can't understand it all. And so, like I said, it gets very complicated. You need to think about what managements in your field you care about the most, and which ones we want to compare. Also, what life cycle stages. Do you care about the birth or the production of that fertilizer? Do you just care about when it's applied, or do you care about what's leaching away and going somewhere else to produce emissions? And the next step is picking which impact categories you care about. So far, I've only talked about one of the many that there are. I've talked about climate change. That's just one of dozens of impact categories you can consider. Climate change is just a function of how many greenhouse gas emissions are produced, and we get some value for its potential to contribute to climate change. But we have other areas too, ozone depletion, if it's contributing to destruction of the ozone layer, eutrophication, and if it's causing algal blooms and dead zones in water bodies and things like that. So as you can see, it gets complicated. So goal and scope is really important. The next 2 steps I kind of lump together, and that's this inventory analysis and impact assessment. And that's where we have a lot of tools out there. So I'm not sure if anyone is familiar with or has heard these words before, but we have OpenLCA. That's one that was mostly used by USDA folks. It's open source, and it's a way to do this life cycle assessment. We also have CIMAPRO, which is the one that I've used the most. It's commercial though, so it costs money. And then we have GREET, which is open access. It's predominantly been used in the context of like fuel and transportation. So these are just a few of the different tools that can do those middle 2 steps of taking all the inputs, turning them into emissions, taking all the emissions and turning them into environmental impacts. So what that looks like is you come up with a list of all the things going into your system, all the materials, the energy, the transportation, everything, and you will put it into some inventory database. And that inventory database might exist in one of these softwares here, or it might just be a spreadsheet, or it might be direct measurements or something that somebody gathered that said, when you burn this much fuel, you have this much emissions coming off of it. And these are 2 of the most common databases, EcoInvent and Gabi, but there are lots of them around. These are just the ones I've seen most commonly. And so these databases will say, based on this input, this is what output you get. And they talk about outputs to the air, to the water, and to the land, so like physical waste. And then the next step is impact assessment. So you need to pick a methodology that you want to use that will translate all of those emissions into an environmental impact. Some of the ones that I've used are the IPCC or the Intergovernmental Panel on Climate Change. So they essentially, for instance, will multiply all of the emissions to the air by some global warming potential to give you the climate change value. Tracy is another tool, and that one has lots of impact categories. They use models that exist, like the IPCC model is built into Tracy, so it's based on a bunch of other models and kind of does a similar thing. And what these give you are these impacts, and those are those different categories I presented. And so these 10 impact categories here, for instance, are what Tracy does. If you use another impact assessment method, then you might have other categories. And you don't have to look at all of those. If you really care about one or the other, then you can pick that one. And I'm suspecting that in the future, a lot of folks are going to have to do this climate change one as we start to think about carbon credits and sustainability in ag. Okay, so the last step is interpretation, where you take all of this information and you make a recommendation to the farmers you're consulting with, or even in your own fields. What are you going to change, or are you going to change anything? And modify as necessary. So here's where I'm going to go into the study that Jason and I and Summit did with a student recently. And so we were using the TAPS program. Who's familiar with the TAPS program? Anyone? Okay, just a couple. But I'm not going to go into a ton of detail with the TAPS program, but it's a multi-state program. It's a farmer competition that allows folks to, in a low-stakes environment, test out different nutrient and water management technologies. That's the big driver there, and test them out and see how they perform without risking it on their own field. And there will be a lot of tools that they can use to, like for instance, sensors and ways to find better ways to predict when to apply their irrigation and how much and things like that. And so they have a number of different decisions that they can make. First of all is, deciding what type of crop they want to do. There's different hybrid selections. I'm not going to go into that today. They also get to pick what seeding rate they want to use. And then most notably for the stuff I'm going to present to you today is the irrigation amount and timing of that, and their fertilizer amount in the form of fertigation events. And they have this, on a center pivot, kind of this block design where you'll be given 3 different plots, and then they'll take the average of all those and scale it up and see who wins, who has the most yield, who makes the most money in these scenarios. Okay. Any questions on that? All right. So the idea that I had when I learned about this program is I was like, this is a great opportunity for us to test this life cycle assessment framework and see what are the environmental impacts for these decisions, because this is a very real application of a lot of different decisions in the same exact location, same soil, same climate. So you're controlling for a lot of things. And so our student, what she did was she looked at only pre-field and in-field. We didn't look at the post-field. And we were just looking at greenhouse gas emissions, so no other impact categories, just climate change. And so in the pre-field side, we were considering production of fertilizers, herbicides, things like that. The transportation of those materials to the field, that was considered pre-field. The production of energy in order to pump our irrigation water, that was all considered pre-field. And then in the field, we also considered the emissions associated with burning diesel for operation of field equipment. We considered those direct emissions from denitrification and nitrification. And then also the combustion of natural gas in order to pump irrigation water for the proportion that was natural gas combustion instead of electricity. And so these were some of the tools that she used. She used SimaPro, which I mentioned is just something that does those 2 middle steps of the LCA. Also used GREET for more of the transportation-based emissions. And then finally IPCC, which was for these direct soil emissions. And what the IPCC does is they just take the amount of fertilizer that's applied, it doesn't matter when, the total amount for that year, and multiplies it by EF, which stands for emission factor. And they have this lovely table where you can put in the default emission factor, which is 1%. So 1% of the nitrogen becomes nitrous oxide. That's their estimate. But you'll note that there is a lot of uncertainty here. That's a huge range. And so this isn't probably the best approach, but it's a good, back of the envelope calculation. Another great thing to see here is that they finally added this. Dry climates has a much lower impact or emission factor, which is great because we were often being compared to, areas in the Corn Belt, and our emissions are going to be much lower than theirs. So we should have that reflected in this emission factor by having it much lower. And so wet climates, theirs is closer to 2%, ours is 0.5%. So at least they're improving there. But for the sake of this student's work, we just used the 1%, just as a baseline. And so this is what we found. This plot shows the different parameters here and the yield-weighted greenhouse gas emissions. So remember the functional unit, I said that we should be comparing these all on an even playing field. We did this on a yield basis. And so if you have higher yields, your net impacts are going to be lower as a result. So you're incentivized for growing better crops. The orange ones here are the pre-field parameters, so all those things that were on the left side in that systems diagram, and then the blue are just the in-field ones. And the first thing that jumps out, or hopefully should jump out to you, is that the infields are contributing the most. And so if you had to choose one of these to focus on, we just should be focusing on measuring things in the field. The pre-field stuff is important, but not quite as important. The other thing is that if we do this analysis in the future, we can probably ignore any of those minor amendments like herbicides and seed production, diesel production, even diesel combustion in the field. That's pretty negligible. So those differences in how the fields are being managed there isn't that significant. But the production of the fertilizer, the production of energy for pumping irrigation water, the combustion during irrigating, and then also the direct soil emissions resulting from fertilizer. That's where we need to focus. And there is a lot of uncertainty here, partially because there's a lot of uncertainty in the estimation methods that we use, but also because farmers were managing their fields differently. And so there's going to be a lot of variability here. And so as a next step, we hope to apply more intense models instead of just this multiply by 1% or multiply by 0.5% of fertilizer, to incentivize using different types of fertilizers, applying it at the right time or the right place, the right method, things like that. And so there are some models out there that are more involved. This is a process-based model, the denitrification decomposition model, abbreviated DNDC. And that's what we've been using extensively in my group. And essentially, you have a lot of inputs that go in. It's all of the climate parameters, which we can get from Mesonet or some weather station, if those are near a farm site. All of the human activity, when the tillage happened, when the planting happened, irrigation, fertilization, dates, times, locations, all of that, amounts. Then what specifics about the crops you're growing, and we're working on ways to calibrate the model specific to hybrids, so that's something that we're hoping will be more streamlined in the future. And then also all those soil properties, which we've heard a lot about in previous talks today and yesterday, about how important those things are. And then the model runs through a number of simulations, like it simulates the crop growth every day, water dynamics, and then nitrification and denitrification are the two big ones that I care about in my work a lot, because those result in nitrous oxide production. And so using this model instead, we can get hopefully better estimates of nitrous oxide emissions. And that's what another one of my students has been working on. She just defended her master's a couple weeks ago. And this is a plot that shows the yield-weighted nitrous oxide emissions as a function of fertilizer applied. And probably what you have all heard is that as you increase fertilizer, you increase emissions. And that is what we saw with both of these models. With IPCC, it makes sense that it was strictly linear. And not strictly, I should say, because these are yield weighted. So if you did better with your yields, then your total yield weighted emissions are going to be lower. But this is just like that 1% function. DNDC though, it's got a lot more going on. It's got irrigation included, tillage, the types of crops. It's also been calibrated to yield, so that takes into account some of the specific growing conditions here in Oklahoma. But the question becomes, which of these is right? And without measurements, we don't know. And so that's kind of the next step that we're hoping to do with this program, is go out there and actually measure them. And this is the method that I used in my master's and PhD. I know Jason's used it quite a bit as well. And it's where you use a static chamber, you place it out in a field, so you'll have a base, just like a PVC tube or some sort of shape, essentially. You place it out in the field. Ideally, you have one in row with the crop and one between row, so you get a representation of the field out there. You'll place the top on there, start a timer, and start collecting samples over time. And then you take those gas samples back into the lab and analyze them on a gas chromatograph. And then that will give you concentrations of that greenhouse gas over time, which can be translated into fluxes. So if you have increasing concentrations, you have a net flux, positive flux from the soil. If it was decreasing, that means the soil is taking up the greenhouse gas, which is awesome, and sometimes happens with methane. But that's the gist of this method. So that is the one here on the left. An alternative, and one that we're exploring more now, is the use of an automated system. So one that you just carry, we call this in my group the backpack unit, because there's a backpack you can wear, and just carry the chamber out and place it down, and within a couple minutes it will tell you the flux immediately. So this could be more real time in the future, and we're hoping to kind of figure out which of these methods is better for different scenarios and things like that. And so the idea here is that we would get these field measurements and use those to, almost fell over, use those to calibrate these models and make them better. Because if we know which model is doing better, then we can use that one. Or if we know a way to improve the model to make it more aligned with measurements, we can do that. So that's the goal as a first step. And then we would want to go in and do the full life cycle assessment. So not just greenhouse gas emissions, but also eutrophication, maybe ozone depletion or acidification, things like that. And looking at more than just the soil emissions, but also those earlier and later life cycle stages. And then we hope to get feedback from folks in this program and figure out what they care about, because I personally care about greenhouse gas emissions, but maybe the participants don't. And so I want to know what is most important for them, and also compare to what's going to be most important in policy in the future, and use those to develop a tool, because right now the TAPS program has something where they can see, for instance, real-time soil moisture readings and try to determine what they want to do for their irrigation decisions. So I'd like to have a tool where they could maybe toggle fertilizer amounts or timings and things like that, and see real time what is their environmental impact score for various categories of interest to them. Because the ultimate goal is that that would be included in the TAPS competition, in addition to the economic side, also including this environmental one, and maybe finding a way to make that part of the economic one, like maybe you're incentivized for emissions reduce. And then teach people about it, like at events like this, and so on. And so that's kind of my long-term goal with this program, is to try to incorporate this LCA framework, also thinking about the economics that folks have already been very involved with for this site. And then there's the social consideration, what do the participants care about, what's going to translate in the long term. Because I can talk about greenhouse gases all day, and if no one cares about them, then my talk was moot and not important. But anyway, you can go to this website if you want to learn more about the TAPS program. And I just wanted to take a moment to acknowledge my research group that's helped with a lot of this work. Got Summon and Jason out there. Thanks for your help with all this TAPS stuff. Some of our collaborators over at UNL for the TAPS program. my good friend Diana Byrne, who's the LCA expert over at UK, and together we got a grant to purchase this life cycle assessment software. And so with that, I just wanted to end by saying I hope that you guys learned something about life cycle assessment, and maybe if you hear it in the future, you'll be able to talk to somebody about it. But I think it's going to be a really big thing moving forward in agricultural systems, and I hope that that will go in a positive way, and that will consider economics, environment, and social sustainability all together. So with that, I'll take questions. Thank you. Sorry, say that again? How long does it stay in the air? Nitrous oxide stays in the air for over 100 years. Yeah.
Any other questions? I got one question for you, Mary. So a lot of times we talk about like carbon sequestration, and I'll talk about that here in a minute, on a per acre basis and things like that. But you point out that you do your analysis looking at the emissions per unit of production. Can you tell these folks why you do that?
Yeah, so, and you might have a different answer to this, Jason, but my answer is I'd like to incentivize the managements that lead to increased yields. And so if you have to, in an area like Oklahoma, apply a certain amount of irrigation to get that yield, you shouldn't be penalized, I guess, for that added irrigation if it contributes to more life cycle greenhouse gas emissions. So we should be normalizing to a unit yield basis. However, that does mean that there's going to be more risk because you don't know going in what the year is going to be like, and if your irrigation applied is going to increase those yields or if it's just a bad year. And so I think that it helps incentivize good practices that yield good results, but there's also the risk there too. Yeah.
Yeah.
I'll turn it off.
All right. And just to give a little context to what Mary's been working on, a couple years ago, we were working on this TAPS project, really trying to get funding for it, if you want to be honest. And the folks in Colorado and Nebraska, they were talking to feed yards and ethanol plants and other animal feeding operations that are consuming the grain from western Kansas and the Panhandle of Oklahoma. And they're interested in trying to reduce their carbon and water footprint, both their carbon and their water footprint. And you guys know why, but we thought we could look at this data and really start to understand what is going on with the carbon footprint per bushel, because that's what they care about, because they're feeding bushels, and if they're not feeding bushels from western Kansas, they're feeding bushels from Iowa. So when you start thinking about all these things, if those bushels are coming from Iowa, then maybe they're not pumping as much water, so they're not generating as much electricity, but they're hauling trains of corn from Iowa to western Kansas. And so there's a lot of things going on that make the work that Mary's doing very important when informing our agriculture and how we can manage. And one thing that's striking from her data is that when you look at it, you're talking about efficiency. If you are trying to maximize efficiency, then you're doing as good as you can really on the carbon footprint. Am I necessarily wrong, Mary? So that's one part of it. And for 10, 15 years, I've been working on this deal, the soil carbon sequestration. And I've been asked to give this talk at the American Association of Crop Consultants. I went to Texas and gave it. I've been asked to go to Nebraska and Kansas, but I told them I couldn't. And so I figured I'd share it with you, because my understanding is a lot of these organizations that are trying to assimilate carbon credits are are approaching crop consultants in some parts of the world, of the country, and asking them if they'll take the soil samples for it. And so let me get into my slides. When I first started at Oklahoma State in 2008, they had passed a legislative action that gave the Conservation Commission the capacity to verify and validate carbon credit programs in the state of Oklahoma, and that's soil carbon sequestration, downhole geologic sequestration, forestry sequestration, anything you can think of. And this was back when George Bush was president, and at that time you had the climate exchange in Chicago where you could trade these credits. I mean, it was a much more transparent time in carbon credits back then than it really is now, because now it's kind of the Wild West, but I don't get into that. But anyhow, we were trying to develop protocols where we could go out and monitor soil carbon. We did a lot of work on farm in the North Canadian River and all over western Oklahoma trying to figure out how much carbon we could actually sequester with no-till management. And I won't get into that a whole lot, but that's where I started, and I've been chipping away on these sampling protocols for quite a little while. And that carbon program, like I say, it provided validation and quantification. And so the validation confirms that management is imposed. So on no-till ground, the plan was to send people, the conservation district folks, out to verify that the crops that were planted into intact stubble. That's the definition of no-till. And then the quantification was the direct measurement or modeling of the carbon accumulation in the soil. And we never got into modeling because I'm not as smart as Mary, so I just went out there and punched a million holes in the earth and analyzed for soil carbon. And the modeling would be good and ideal, and I think a lot of these organizations that are getting into this game are going to try to use modeling. But the interesting thing is when we're done with this presentation, you might question as to whether or not those models are fully informed of how much carbon is accumulated, because I'm not so sure that we've been measuring it right. So variations, so a few challenges with measuring soil carbon that we'll discuss are variations in calibration of laboratory equipment, temporal and spatial variations in soil carbon, similar to what Reynilda talked about, differences in how roots can be ground into soil samples, and I didn't get, Reynilda has a chapter on that, and I didn't get that in the presentation, but how we manage roots in the soil sample and how that influences the final carbon number that we get out of our machines in the labs, and then the bulk densities and how it changes with organic matter increases or even with soil moisture. So then you have measured organic carbon can be impacted by what phase of the rotation you have. We got data on that. You can have samples after harvest or after termination of cover crops or before tillage or after tillage, and all that matters, especially if you can't get a good handle on what's going on with the roots, because those roots, whether they're dead or alive, can influence these numbers. And I don't know what to do, like how to approach these problems that I'm going to discuss, but I know that all these problems matter. And so if you want to approve cover crop sequesters carbon, for example, you go right out after termination, and that's when you're going to have the most carbon. But if you wait 2 months after you've planted a cash crop, and it's just getting going, or even 3 weeks, you're going to have lost carbon from that system because those roots would decompose. And I think more fair assessments would be to sample after harvest of the cash crop when everything is equal. And when you do that, you tend to find less effect of that cover crop on the carbon. So when do we sample? I think this is some data. This come out of Goodwill, and this is where I first observed the impact of rotational effects. And so in this rotation, so we had sorghum stalks, we had wheat stubble, we had growing wheat in these plots, and then we had those minimum till with sweep plow, and then no till. And if you look, pretty much all these numbers are the same, except this one right here is significantly higher. This is the organic carbon in that soil, and then there wasn't really any difference among these other treatments, except the wheat stubble had the least carbon in it, but it wasn't really significantly different than any of the others. But this high carbon number in these sorghum stalks is a direct result of kochia infestation at the highest level in those plots. Those plots, that particular treatment, for whatever reason, that year in that rotation had a heavy load of kochia. And what we're measuring is the root mass of that kochia. And so that's where I start to observe and think about how rotation affects things. As far as the calibration of equipment, and this is something that got real sensitive to me because when I sent 1,000 samples down to Highland's lab, you could see every day they calibrated that because they were shooting for calibrating it plus or minus 5% around the known value for their standard. And what happens is 5% on a 1% carbon value is a range of say the average, where is it? Yeah, about around 1% is, you can have 1, or you can have values that are 1.05, or you can have values that are 0.95, and you're well within the calibration of that machine. And that's pretty damn good. But over here, when you equate that to metric tons of CO2 equivalent, which is the units they use for carbon sequestration, that error between 1.05 and 0.95 is nearly 3 metric tons. And for perspective, we assume that you're going to sequester about 0.2 to 0.4 metric tons per year under no-till management in the state of Oklahoma. So that's how much air, and that is in a depth of soil that is 6 inches deep. Does that make sense? So the way we corrected that is we would run known checks every 10 samples with these 1,000 samples. So we'd run 100 checks and then post-calibrate their calibration to where those checks were the same number every time. And I don't know if that's right, but that's what we did. And so we were pretty confident in our carbon numbers and at least the reproducibility of it. Another thing is this, we usually see an accumulation of carbon in the top 4 inches. You look at some of the data that Brian's collected from the long-term studies, and it's actually very highly concentrated near the very near surface of the soil. But this is a 4-inch depth, and then you have going down to what, 2.5 feet, I think. But all your accumulation of carbon occurs right up here in the surface, and you really don't get much change at depth. And so we have to measure the depth to some depth, because what we observe is that you do get changes over time in the profile carbon, and I'm not sure why, but I know these are real changes because we spent a lot of effort making sure that we were calibrated over time and that we could monitor this stuff pretty accurately in the lab. But what this data is this was no-till ground that was sampled in 2009, and then no-till ground that was sampled, the same ground that was sampled in 2012. And you'll see that at the surface, we still have a little bit more carbon than, 3 years later, but then the profile lost carbon. So the net effect is that we lost carbon from this profile. And if you remember, from 9 to 12, we were living pretty large prior to 2009, and then we went into a drought, and we were right in the middle of it, starting to pull out in 2000. Well, we were right in the middle of the drought in 2012. And so that's the effect of drought. And you'll lose carbon across the landscape during drought because you're not generating primary productivity and you're not putting roots in the ground, you're not adding carbon to it, but you're getting some decomposition. And now we lost more carbon from the conventional tilled system, but this is a problem if you're planning on monitoring only those fields that have been had no-till or grass plantings, because you go out there after a wet cycle and you sample, and then you go out there after a dry cycle, the net effect is that you've lost carbon and you're going to have to pay somebody money back. Does that make sense? If you're basing your carbon contract on soil sampling. So organic matter is less dense than minerals, and so the bulk density will actually go down if you're accumulating carbon. So that's another challenge. You can't just go out and take a soil sample and have it analyzed for carbon. You've got to get the bulk density, and you've got to get the bulk density, in my opinion, of every sample you collect, because the carbon variance will affect the variance in the bulk density. So if you only take one bulk density number, then you're going to have error in that relative to the carbon, and it's kind of a problem I won't get into. Now we can also see that the gravimetric water content, and this is just the water content versus bulk density, has a somewhat predictable impact on the bulk density. So carbon content affects bulk density, compaction affects bulk density, and a water content affects bulk density. So you can't just take bulk density once, even if you took it from every sample, and then assume it'll be constant through time. You have to do it every time, is kind of the moral of that story. And that's kind of the same slide. And it's actually stronger when you get down into the 2 to 3 foot depth.
Right, I'll go back and say why we need to get bulk density off the sample we collect with the soil probe.
Oh, yeah. So this pertains to a call I got from a grower over in Creek County. And so this guy went out and take a soil sample, and somebody had told him if he wanted to monitor carbon, he just needed to send it to a lab. And I won't name the lab. I would, Brian would. But the lab analyzed it for carbon and a bunch of other stuff and bulk density. And if you know anything about bulk density, is a bulk density of 0.7? What's that mean? Does anybody know? If you walk on a soil with a bulk density of 0.7, you will make deep footprints because that's nice, soft, supple soil. So I asked the farmer, I said, what kind of, man, this is pretty good soil. You got a bulk density of 0.7. And he says, well, I had to have a hammer to get the shovel in the ground. And I was like, well, then your bulk density is wrong. Because if you got to have a hammer to get a shovel in the ground, your bulk density is no less than 1.4. But this company, I don't know what happened there, but this outfit told him that he could send the sample, and what they did is they took that crumbled sample and they poured it in a graduated cylinder to get the volume of it, and then they took and dried it and determined the dry weight, and they took that dry weight and divided it by its volumetric volume and that's not the way you take bulk density. The way you have to take bulk density is you drive a probe into the ground, and then you push the soil out of that probe, and then you meticulously cut it to a specific depth so that you have the depth, and the cutting diameter of the probe determines the volume. And that's how you get bulk density if you want to monitor carbon. But that was striking to me, and one of the reasons why I'm giving this talk is that there's a lot of people out there in the world telling a lot of people how to do things, and it's wrong. And if it goes to a court of law, most of the stuff that I've done in my career on monitoring carbon would not stand up in a court of law, especially if you brought me in as an expert witness. And that's a serious deal when you're talking about the dollars that people are throwing around, and they're kind of very few dollars per acre. But if you've got a big farm signed up on this, and 5 years later they tell you it's no good, and you have to come up with that money because you didn't read a fine print contract, then you might have some trouble.
File? You got shot.
Warren GHG something. But to keep us so these are things that run in my mind all the time because I get these calls, and I don't know if you guys have these discussions with growers or not. But I don't remember what was next on my slides, Brian. Trying to remember what was next so we can keep going. Yeah, we covered this, covered that. I think we're close to the meeting. How close are we, Brian? Because have y'all had any conversations with anybody about this? What? If you get it going, Brian, I can get us to where we were. I think you have to enable edit. Yeah. So that's kind of the bulk density stuff, but this is how we measure it. And like Sumit Sharma's master's degree was comparing these 3 probe types, and what he found is that we did increase the bulk density a little bit when we use that small hand push probe because of the diameter of it, and that's generally when your soils are kind of moist, you can get a little bit of air. But then he also developed or evaluated some protocols for calculating the carbon content, and generally you used what's called a fixed depth. So you go to a fixed depth of 12 inches and you analyze that and get the total carbon in that 12 inches. But you can also fix the mass, because that mass will vary with bulk density in that top foot. But then if you say, well, I'm going to just calculate carbon mass on a mass basis, a carbon mass per soil mass basis, and you keep the mass of soil constant and vary the depth, and you can do that mathematically, then you get the same numbers regardless of the probe type. So that's one win that we found in our efforts to try to develop a uniform and consistent way of measuring carbon. And I won't go into the math on this, but this is a traditional way of, you'd analyze each one of these depths, multiply it by the bulk density to get carbon content. Over here, we're truncating that bottom depth and using a fixed mass that's equivalent to the lightest soil that we had. And so we get more consistent numbers. Another thing about it is spatial variability. So we had a landowner up in Noble County ask us to go out and do a baseline carbon evaluation of this farm. And you can see it's Swiss cheese with respect to the soil types. This thing has got everything from a deep port silt loam to this crappy Norge up here. And we went out there and took 4-foot cores off of 18 locations. And what we found is that the carbon mass, what is this, to 12 inches ranges from 47 to 70 metric tons. If you go to 3 feet, it ranges from 83 to 200 metric tons. And that's a hell of a lot of variation in one field. And so how are you going to monitor that and measure that? Even if you're just in the top 12 inches, you're almost doubling the carbon. And so we with the carbon program, we decided we were just going to monitor distinct specific locations in the field and try to, instead of trying to determine the amount of carbon accumulated in every field because of that problem. And then when you look at Oklahoma in general, this is Texas County, Ottawa County, Kiowa, Stevens, McCurtain, and Payne County. And on the right-hand side of each one of them tables is the metric tons of CO2 equivalent for each one of these different soils. You can get this off the soil survey. You can calculate it from data on the soil survey. But in Ottawa County, it ranges from 40 to 270 metric tons. In Texas County, it ranges from 60 to 90. And these are just randomly selected soils. But see how much variation we have due to the inherent differences in soils across the landscape. Like this verdigris, anybody deal with a verdigris clay loam? That's the black dirt you see up in Ottawa County, but it's got mountains of carbon. You can find that same kind of carbon if you go up around Ponca City and like north of Ponca City, and they've got that real black soil. That's kind of number you'll find there. So the question then is, how many samples do you have to have? And all that variance, both large scale and small scale, affects the number of samples you need to quantify carbon changes over time. And so Sumit analyzed his data with a power analysis, and I won't go into that, but it essentially determines how much degrees of freedom or how many samples you need to overcome the variance in a data set. And so if we expect to sequester 0.4 of a metric ton per year, and the standard deviation found in these 47 locations that Sumit used in 18 different soils, and samples were collected within 300 square feet, and we were only looking at 12 inches, the number of cores required ranged from 10 to almost 660 samples with an average of 115. And that's in a 300-foot square area. We would need an average annual sample collection within that small area of 115 cores. So then we decided we were not going to do that, and we started looking at looking at multiple years worth of accumulation. And so you figure you wait 4 years and that 115, this is a little bit different math. Number of sites assume 10 cores per site, and we assume 80% confidence that if we had 13 intact sites, we could measure a significant increase of 1.6 metric tons in 4 years. And that's where we're taking 130 samples per observation from 10 sites. So we take 12 or about 13 cores per site, if that makes sense. But the long and short of it is you're going to have to take a lot of soil samples. And you're going to have to take bulk density and you have to analyze them all for carbon. Another thing that we don't even touch on is carbonates. Our soils are loaded with carbonates. Despite the acidity of our surface, the subsoil is loaded with carbonates. You go to the panhandle, they're loaded with carbonates from top to bottom, and we don't even worry about it. We take acid and we burn that off and we get the total organic carbon. But what I wonder is we've eliminated all the acid from the atmosphere through scrubbing sulfur out of the coal and oil and everything we burn. And so we're no longer acidifying the planet like we were 30 years ago. And so I don't even know what's happening with carbonates because they're no longer being sent in the atmosphere through the acidification. And I'm not so sure they might be accumulating again now that we've removed all that acid from the atmosphere, and we have no idea. So summary, collection and analysis of organic carbon stocks is not simple. It's not easy. It must consider variations in bulk density and carbon concentrations and changes over time. Must overcome variability in order to measure a small change in a relative large pool. I'm not sure how to handle the roots, and we didn't even get into that. I don't know how to handle the carbonates. I don't even know if they're changing. We can use a fixed mass method to reduce the bulk density error, so there's a shining beacon of hope. But when you get approached, if you do get approached for sampling carbon, or you talk to people about sampling carbon, these are the things that you're talking about dealing with. And nor the USDA, the Soil Health Institute, or any organization that I know of has put forth any kind of protocol that would get past me in a court of law. And so be thoughtful about that if you ever get into this space. But that's my spiel.
So, any questions for Jason? Because I know I do. Mike's got one. You always count on Mike.
Yeah.
So you're trying to tell us on taking your core samples that we probably ought to get GPS coordinate of where we took the core and come back every year to that same spot?
Yeah, that's the only way that's rationally, that's the only way you could reasonably do it. I don't really know, like right now, I don't know what carbon's worth. The federal government has not put legislation in place that requires us to manage carbon. And so everything you see outside of California and a few other states and Canada, that's all voluntary effort to reduce their carbon footprint, to try to go to zero net emissions for all these companies. And it's pretty shadowy. And does that make sense? And so we don't know what the standard is and the expectation for accuracy or precision is, because there's no federal guidance and everybody's just doing their kind of own thing, trying to sell their wares as best I can tell. But if you wanted to do it correctly, and most efficiently, is you would monitor a single location in multiple fields instead of trying to monitor the entire field. And like I know there's companies out there that are doing discrete specific point sampling and then trying to calibrate models for that. But I still would like to see the way they analyze the data, or the soil samples specifically, and how they handled certain sample collection and processing things before I'd even believe that the samples that are taken are informing the models correctly. But there is the soil science community come up with this stuff 30 or 40 years ago, and they've never addressed these things. Does that make sense? And so I'm out of it because I'm an administrator. I'm dead weight. But you guys are going to have to deal with it.
Has anybody been approached?
Your results, and you do what
James?
you do to
Yeah.
come out with the result you want.
Oh, yeah. I could make I could make.
Do you have a question, comment, or just admittance?
Yeah. Yeah. Yep. Yep. Yeah.
Hold on. You've got a lot to say.
There's a lot of programs out there. Some of them are say they can measure it. Others are just paying for the practice. For instance, Oklahoma Association of Conservation Districts, I believe, I don't know how widespread, but I know that they did or do have a program where they're just paying for the practice. So that's your soil health
Yeah.
things,
No,
cover
and
crops,
damn, I'm about to fall off the stage. But what you just said is completely accurate. I appreciate it because when we get paid for practice, I'm comfortable. And I think that we are doing good by accumulating some amount of carbon in the ground. What my nerves get anxious is when we are asked to validate
Right.
and
Right.
quantify the amount that we're putting in, because just like you said, you have temporal and spatial variations that we have not developed sound, uniform protocols for measurement. So you're exactly right.
and if we wanted to, we could even do this here in Oklahoma. Like, it depends on what their measurement is and whose program and how much they're paying. I'll plant nothing but C4 plants, to be honest with you.
Yeah.
If that's the case, I'll just plant C4s.
Yeah. And one thing that makes me, because I asked the NRCS a couple days ago, or about a month ago at their state technical meeting, because they're moving all this climate smart funding, and we're going to see a lot of grass plantings and a lot of irrigation efficiency improvement money coming out of that. I think that'll be good. But my anxiety, because I had a student she sat on the library lawn and protested against climate change as an effort to get the ball rolling to get people to move forward on addressing that problem. And if she ever becomes in a position of influence and sees what we've done and we haven't done anything, and I'm not saying agriculture, I'm talking about society in general. I won't get into my opinions about agricultural role in this unless you really want me to, because we need to be concentrated on food and fiber, and I'll just leave it at that.
Water quality.
But if we're asked to help in this improvement, then what we are asked to do should be valid and impactful on what they're trying to improve. And I'm not so sure that some of the things they're telling us to do.
But for the same reason, you're right about climate smart. From there's so many dollars coming at this soil health stuff that it's.
Yeah.
And so it's whether through us at the state level with the Conservation Commission or whether it is through NRCS.
Yeah.
Seriously, without carbon getting involved yet, at least at the state level through the commission, I promise you that.
No.
I'm leading a program through General Mills up in north central Oklahoma.
Yeah.
I had to get these things board approved in every county district that I have this going in.
Yeah.
The first question they asked me is carbon a part of this? Because if it was dead on arrival.
Who said that? The dead on the arrival part, the boards?
Yes.
Oh, okay. Well, and like I say, I think that we are doing good, like with the projects you guys are doing, and I didn't mean to get into that because I've talked to Dr. Trey Lamb about some of this stuff.
Yep.
Because we're the only state in the country that has a carbon program where we could help growers work with aggregators to provide that validation through the Conservation Commission, the districts. And that's how I know about this. I'm one of the few state specialists in the country that has actually went through this, but we did it 10 years ago, and now it's reared its head again. But nobody wants to work with the Conservation Commission because of the requirements they have, really.
Well,
I mean, and as far as aggregators and corporations trying to do this,
at the same time, I have fewer rules than NRCS, so it's kind of a double-edged sword. But again, there's so many dollars coming at this from all different angles. At the very least, with producers, just stop in your county district offices and ask a question, because there's probably enough dollars. There's a program and a payment to at least help you somewhere, whether carbon's involved or not.
Yeah. No, you're absolutely right. Thank you.
Thanks, James.
I just get anxious about the soil sampling. Like all the payment for practice I'm good, and I think we can do a lot of good with that kind of stuff, but it's when we are asked to take soil samples, there's a real specific way it needs to be done for it to actually be valid.
Any other questions for Jason, formerly known as Dr. Warren? You don't have the title anymore. Thank you, Jason.